Welcome to the flagship studio at the 2024 JP Morgan Healthcare Conference. I'm joined by CEO of Profound Therapeutics, John Lepore. John, thanks for joining us.
Great, Joe. Thanks for having me. A lot of fun to be here.
So let's start by talking about the company broadly. Can you tell me about Profound and its mission?
Yeah, the mission really is to identify novel therapies for patients. That's the starting point. And what's important as a background is that essentially every drug ever made has been targeting proteins, because proteins are the building blocks of life. They're what control normal functions, processes, and by intervening on them, we're able to change the course of disease and help patients. One of the challenges is that there's a finite set of proteins that all the companies have really been chasing in different ways. And I've, as you know, recently joined Flagship from a large pharmaceutical company where I was keenly aware of this problem of always being in competition. And one of the things that's extremely valuable both for patients and for companies and investors is to work in novel space where you could potentially have a first-in-class medicine. So the principle behind Profound Therapeutics is to identify completely novel proteins that no one else knows about. And those proteins either could be drugs themselves to be administered to a patient, or the protein becomes a target for a drug and really, again, open up important opportunities both for patients and for investment.
So maybe this is a good time for a brief history lesson. If we look back to the completion of the Human Genome Project, we were surprised by the finding that the human genome only encoded for 20,000 proteins. That's much less than we were expecting to see. So can you tell me about how the researchers came to that conclusion and how Profound's efforts to pioneer the expanded human proteome are changing the paradigm?
Yeah, it's really interesting. I mean, first of all, the Human Genome Project was just a foundational event in modern science that really underscored and enabled the development of many important drugs. I don't want to undermine it in any way, shape, or form. But they necessarily, at the time, because of the constraints of what we knew about genes and RNA and proteins, had to put some rules in place. And those rules, in retrospect, ended up being relatively conservative. And for example, they defined proteins as being at least 100 amino acids. They defined that each protein had to be from a message that had a very specific start codon. And all of the proteins had to have some degree of homology or likeness to known proteins. And putting those features in place really limited the scope to 20,000. That represented about 5% of the human genome, of the DNA sequence. And it's not surprising that human activity, human emotion, human biology, human disease is much more complex than 20,000 proteins. And it begged the question, well, what's the other 95% of the genome doing? And that's what we're systematically going after.
Excellent. And so, this work with the Human Genome Project, it was completed more than 20 years ago at this point. And so, now with Profound exploring and expanding our understanding of the human proteome, what has changed in that time that's allowed us to get to this point where Profound is now?
Yeah, I'd say there's really the evolution of three related but distinct technological advancements that's enabled us to do what we're doing. The first is how to find novel proteins. And in the past, people have used mass spectrometry to find proteins, but that technology is limited to find proteins that are pretty small, like less than 100 amino acids. So a lot of false negative rates. What we've done is perfect a technology called RiboSeq. And what that allows us to do is to look precisely at the cellular structure called the ribosome, which is where mRNA is translated into proteins. And we isolate those mRNAs, and we're able to identify the proteins actually in the process of being translated. And that's allowed us to see the full universe of proteins, not encumbered by those rules from the Human Genome Project. And what we found so far is literally tens, if not hundreds of thousands of novel proteins that are encoded by parts of DNA that we used to call junk DNA or regulatory DNA because we didn't know what to do with it. But in fact, it's coding DNA and makes these small proteins. And now we're really at scale in an industrial way querying these proteins for what functions they have that might be important as drugs or drug targets.
And you're taking all these technologies and you're putting them into what you're calling the Profoundry platform, right?
Yeah, thanks for the prompt. So the first of the three technologies was the RiboSeq to find the proteins. The second technology, and I think we could call it a technology, is the availability of vast data sets that inform this. Some of the data sets we've generated, but also others have been the product of large consortia with scientific organizations and academic institutions. For example, the UK Biobank, AlphaFold for proteomics, GTEx for mRNA sequences, and there's vast databases that we've ingested and we've put in a searchable database. So as soon as we found a novel protein, we could put it in context of what's known about the biology of mRNA, proteins, gene regulation, and disease. And by doing so, we're able to rapidly, no one pipettes anything, not in the lab, in silico, really get a good sense of which of these might be important drug targets. So that's the second. And the third is one that I think is particularly relevant to the flagship ecosystem, which is the ability to use artificial intelligence and machine learning to inform all of this. Because what we're finding as we identify and describe the function of one of these novel proteins, we could start to put the data into models that predict the function of other proteins. So this is sort of a flywheel of information that allows us to very rapidly identify the most promising proteins.
It sounds like there's a lot of integration that's happened. You know, the company launched back in 2021. A lot of work in the platform. Can you tell us what else the company has achieved in that time?
Yeah, so maybe I'll describe the process a little bit more because that can illustrate what we've achieved. First off, there was the identifying of proteins, and as I said, we've identified tens and hundreds of thousands of novel proteins. The second was to use this in silico database to inform them, and from that we've gotten I would call it collections or lists of proteins that have very important features. For example, proteins that we predict to be on the membrane surface of tumors, or proteins that we predict to be secreted from immune cells and be novel cytokines. So we've got a whole library of these potential novel biologies that are of interest. That's a very significant achievement. The third component we didn't talk about so much yet is once we, through the in silico engine, identify these interesting potential features, we then test them in the laboratory. But instead of doing that one by one, the way the field has historically done it, we're doing them in hundreds and thousands at a time. And these are what we call high throughput systems. So, you know, assays and experimental systems that allow us to look at function in immune cells, function in skeletal muscle cells, effect on fibrosis, and learn a lot that's translationally relevant for disease context rapidly. And then it's only the most important ones that we bring forward as drug discovery programs.
So it sounds like there's a lot to come. Can you walk us through, you know, what you expect and what we can expect to see from Profound in the coming years?
Yeah, look, just to backtrack, I took on the challenge of leading this company because as a physician scientist, what really drives me is to find new ways to help patients using technology. And I saw this as just an incredible opportunity to open up a whole new world of drug targets at almost unimaginable scale, like hundreds and thousands of new drug targets. And what we'll see is rapidly driving towards bringing forward those drug discoveries into clinical programs. So over the next year or two, we'll be making antibodies and advancing programs towards helping patients as rapidly as possible in the clinic, and a really exciting opportunity.
Sounds it. So tens of thousands of new proteins and potential targets for potential new drugs for patients, and a lot of progress to come.
And across almost every indication imaginable. So really a gold mine in all senses of opportunities.
Sounds great. Thank you so much for joining us, John.
Thank you, Joe.